Hello, this is Matt Dean with A Plus College Ready, and today we're going to talk about dissolved oxygen and primary productivity. There's a misconception that aquatic organisms breathe something different than what terrestrial organisms breathe. First of all, you need to make sure that you know that aquatic organisms breathe exactly the same oxygen that terrestrial organisms breathe, and that's the molecule O2. When in water, the oxygen is dissolved, and it's referred to as dissolved oxygen, or DO. The DO concentrations in water are much lower than the oxygen concentrations in the air. And that's why terrestrial organisms with lungs, can't breathe when they're underwater. There's just not enough oxygen in there. There is some, but not enough to support large, active terrestrial organisms. In freshwater, the DO concentration can vary from about 1 up to about 20 milligrams of dissolved oxygen per liter of water. Several factors affect the amount of DO in the water. One of those is temperature. As temperature increases, the amount of dissolved oxygen tends to decrease. Think of it almost like a soda. That if you heat up a soda like a Coke, the carbon dioxide inside that soda will start to bubble out. If you were to leave a closed can of Coke in a hot car in the window, it might very well explode because a bunch of the carbon dioxide that's dissolved in the water will come out of solution and build up pressure. So as temperature goes up, dissolved oxygen levels go down. Another factor that affects DO concentration is movement. Moving water, as movement increases, let's put it this way, as the movement of the water increases, the amount of dissolved oxygen in that water also tends to increase. Rivers, streams, are going to have more dissolved oxygen than like a lake or a pond. Salinity is another factor, saltiness. As salinity increases in a body of water, the amount of dissolved oxygen in that body of water tends to decrease. So more salt, less dissolved oxygen. And finally, the organisms that are present in the water. If there are a lot of photosynthesizers, so more photosynthesis should mean more dissolved oxygen because photosynthesis produces oxygen. On the other hand, if there are a lot of animals, a lot of other organisms doing lots of cell respiration, as cell respiration rates increase, the amount of dissolved oxygen in the body of water is going to decrease. So now we want to talk a little bit about primary productivity. Whenever you hear the word productivity, or maybe produce, or producer for that matter, any form of the word produce, you want to think about photosynthesis. Productivity has to do with photosynthesis, the amount of organic molecules built or made during photosynthesis. So as my definition here says, primary productivity is a term that describes the rate at which plants and other photosynthesizers like algae and cyanobacteria produce organic carbon-containing molecules through the process of photosynthesis. Now we might oftentimes talk about gross productivity and net productivity. Don't let those terms confuse you. Gross productivity is the entire photosynthetic production of organic compounds. For example, if you have a job, you know that your gross pay is the total amount of money you earned. So you take your hourly wage times the total number of hours that you worked. That's your gross pay. Now your paycheck is not actually made out for that amount because before your paycheck is written to you, deductions are made from that paycheck, things like taxes and Social Security. The net productivity, on the other hand, is a term that refers to the organic compounds that remain after organisms have used some of the organic molecules for their energy needs. They've used those organic molecules to do cellular respiration. So your net pay, keeping my analogy going here, your net pay is what you actually get. It's what you're left with from your gross pay after deductions have been made. So in this case, think of cellular respiration like a deduction. The photosynthesizers make a certain amount of organic molecules, and then some of those organic molecules are used up for the purposes of cell respiration. Some other important terms to think about. A lot of people think that plants do photosynthesis and animals do cell respiration. Well, that's sort of right. Photosynthesizers do both photosynthesis and cell respiration. Non-photosynthesizers do only cellular respiration. Cell respiration is occurring all the time. It's occurring 24/7, 365. You need ATP continuously. Your body is always making ATP. Cell respiration doesn't stop. Whereas photosynthesis can't happen for long periods of time in the dark. Photosynthesis essentially will shut down in the dark. Since oxygen gas is a product of photosynthesis and it's also a reactant for aerobic cell respiration, dissolved oxygen levels are a good way to help gauge the primary productivity that's happening in an aquatic environment. So on the AP exam, you might be asked to calculate some factors related to primary productivity. And one of the things that we can calculate is the net primary productivity. And we can do that by measuring the changes in the dissolved oxygen for a sample of water. Let's assume there's some photosynthesizers in that water, maybe some algae. And let's incubate that water in light. You've got to remember that in the light, both photosynthesis and cell respiration are happening. Now, if we're in bright light, probably photosynthesis is happening faster than cell respiration. So we can calculate the amount of DO consumed by cell respiration by measuring the change in dissolved oxygen for a water sample, again with algae, but this time only incubated in the dark. Remember that in the dark, only cell respiration occurs, while in the light, photosynthesis and cell respiration are both occurring. So we can use this equation down here at the bottom to help us out. The net productivity, the net amount of carbon fixed or organic molecules produced, is equal to the gross productivity minus organic molecules used up by cell respiration. We might rearrange that equation for GPP, for gross productivity, and we might say that GPP is equal to the net primary productivity plus respiration. So let's look at some actual problems and how this might work. But the key thing is we're using what's called the light-dark bottle method. In the dark, only cellular respiration is occurring, so the amount of oxygen should drop since aerobic cellular respiration uses oxygen. In the light, both cell respiration and photosynthesis are occurring at the same time. Typically in the light, photosynthesis is going to happen faster than cell respiration, assuming the light is bright, and therefore we should get an increase in dissolved oxygen. That increase, though, is not the total amount of oxygen made, because some of the oxygen was also used up at the same time. So let's see how that works in a problem. Before we get to a problem, let's think about a couple of more topics. As dissolved oxygen is produced by photosynthesizers, carbon is fixed or assimilated into the organism's organic molecules. So as plants do photosynthesis, they're releasing oxygen, but they're also using the carbon dioxide that they took in during photosynthesis to make sugars, to make other organic molecules. We call that assimilation. We might call it the fixing of carbon. We can calculate either the net or the gross amount of carbon fixed using the formulas given to us at the bottom of the screen here. So from these formulas, these two you just need to remember. You need to memorize these two. They're not on the AP Bio formula sheet. But the two down here at the bottom are. And they're essentially conversion factors. So let's say that we wanted to calculate the gross amount of carbon fixed in a bottle. What we would first want to do is calculate the GPP in terms of oxygen. So we would use this relationship. We would solve it for GPP. Like so. And we would calculate the milligrams of oxygen produced per liter of water. One conversion factor to be familiar with, very oftentimes when we're talking about dissolved oxygen, it's going to be measured in what's called ppms, and those stand for parts per million. 1 ppm is the same as 1 milligram per liter. So don't let that confuse you. 1 ppm is the same as 1 milligram per liter. So let's say we've calculated a gross primary productivity, and we've got that that calculation in terms of milligrams of oxygen per liter. We can then take these two equations down here at the bottom of the screen. We can first run our number through the top equation. That top equation essentially converts milligrams of oxygen per liter of water to milliliters of oxygen per liter of water. So it's a conversion factor. Then we're going to take that answer, we're going to plug it into the second equation. So we're going to use this equation now. We're going to take our answer from the top equation, plug it in right there, multiply that by 0.536, and again, that's a conversion using the balanced equation for photosynthesis, which will convert from the amount of oxygen released to the amount of organic molecules made, or in this case, the milligrams of carbon fixed per milliliter of oxygen. All right, so we'll end up with milligrams of oxygen fixed per liter of water. So let's see how that works in a real problem with real numbers. So problem 1 here. Water with algae inside is placed into a completely dark environment. Remember that in the dark, only cellular respiration can occur. Photosynthesis can't happen there. So our oxygen levels should drop while the bottle and the water are in the dark. If the initial dissolved oxygen concentration of the water before it's placed into the dark is 12 milligrams per liter, and that's milligrams of oxygen per liter of water, and the dissolved oxygen concentration after 1 hour in darkness is 8 milligrams per liter, how much oxygen is consumed by the plants per hour? So this is a straightforward problem. We're starting out with 12 milligrams of oxygen. Again, we're starting out with 12 milligrams of oxygen per liter of water. So we've got 12 milligrams per liter. That's how much DO we're starting with. After an hour, we only have 8 left. So if we subtract the amount we started with, if we subtract from the amount we started with, the amount we're left with after an hour in darkness, we end up with 4 milligrams per liter of oxygen. So that's telling us that the organisms in the water used that much oxygen in 1 hour. We're going to label that value as R, the amount of oxygen consumed by cell respiration. We'll come back to that R in a minute. Question 2. What process or metabolic pathway consumes this oxygen? This past year on the AP exam, it was pretty obvious that a lot of students didn't know what the word metabolic pathway meant. A metabolic pathway is a chemical process. Usually it's a multi-stepped process. It involves multiple enzymes that carries out some function in an organism. We might think of the Calvin cycle as a metabolic pathway. The Krebs cycle. We might think of cell respiration itself as a metabolic pathway. We might think of digestion as a metabolic pathway. In this case, the process that's using up that oxygen is aerobic cellular respiration. So I'm just going to write ACR, aerobic cell respiration. Question 3. A bottle of water from the same source as the bottle from number 1 with algae inside is placed into a sunny environment. So again, when the bottle's in the sun, both photosynthesis and cell respiration occur. If the initial dissolved oxygen concentration of the water is 12 before it's put into the light, and the concentration after 1 hour in the light is 25, how much net oxygen is produced? So to calculate the net amount of oxygen, we're going to take the amount of oxygen that we ended up with, 25 milligrams per liter, and we're going to subtract from that the amount that we started with, 12 milligrams per liter. So our net amount of oxygen made is 13. Cellular respiration photosynthesis rather, produced 13 milligrams of oxygen per liter of water more than it used for cell respiration during that 1 hour. So question 4, and we have 2 question 4s here. The first question 4, what process or metabolic pathway produced this oxygen? Photosynthesis is a metabolic process. It's a multi-step chemical process involving multiple enzymes. So the answer to this one, photosynthesis. Part 4 wants to know how much net carbon is fixed during the same period. Alright, so to calculate the net amount of carbon fixed, we're going to take our net primary productivity in terms of oxygen that we calculated in the previous problem, 13 milligrams of oxygen per liter of water, and we're going to use that with these equations or conversion factors from the AP Biology formula sheet. So we need to start with the milligrams of oxygen per liter. So we're going to take 13 milligrams of oxygen per liter. We're going to plug that in to here. So we need to multiply that by 0.698 milliliters per milligram. And what that gives us, that essentially converts milligrams of oxygen per liter to milliliters of oxygen per liter. And once we do that calculation, we get 9.074. And that's in milliliters of oxygen per liter of water. We're now going to take that answer and we're going to plug that in here to the second equation. So we're going to take 9.074 milliliters of oxygen per liter of water. We're going to multiply that by a conversion factor, 0.536. And what that's going to do is that's going to convert, using the balanced equation for photosynthesis, the amount of oxygen released by photosynthesis, it's going to convert that over to how much carbon was actually assimilated or used to build sugars and other organic molecules in the plants. So when we multiply that through 9.074 times 0.536, we end up with 4.86. And that is milligrams of carbon fixed per liter of water. That's the answer we were looking for. Now if we go back to the previous question for a second, we found on the previous screen that the amount of oxygen consumed by cell respiration was 4 milligrams of oxygen per liter. We also calculated the net primary productivity in terms of oxygen to be 13 milligrams of oxygen per liter. The last question is we want to calculate the gross productivity in milligrams per liter of oxygen. Remember the equation, NPP equals GPP minus R, or if we solve that for R, GPP equals NPP plus R. So we have an NPP of 13, we have an R of 4, so if we take our net primary productivity, 13, plus our oxygen used up by respiration, 4, our final answer for number 5, for GPP, gross primary productivity, is 17 milligrams of oxygen per liter. So we're saying that photosynthesis produced this much oxygen. Now we're saying at the same time, the organisms in the water used up 4. And therefore we only saw a change in the light of 13. So 17 milligrams of oxygen were produced. At the same time 4 were used up. So that left us at 13 to the good. 13 more than the plants used. Alright, so here's a slightly different type of primary productivity calculation. Samples of water, all from the same source, were incubated at varying light concentrations. Assume that all the bottles contained an equal amount of photosynthetic organisms and that they were all incubated for the same amount of time. The dissolved oxygen concentrations for each bottle are given in the table on the next slide. Use those dissolved oxygen values to calculate the gross primary productivity, the net primary productivity, and the gross amount of carbon fixed for each sample. Alright, so here are our data. We have an initial bottle that started out with a dissolved oxygen concentration of 8.4 milligrams of oxygen per liter of water. We then put it in the dark, and after a period of time in the dark, the dissolved oxygen concentration was 6.2. So if we take those numbers and subtract them, let's take our initial 8.4 minus our dark reading, remembering that in the dark, only cell respiration was taking place. So if we subtract those two, we end up with 2.2. That 2.2 we're going to label as R. That's the amount of oxygen consumed by cell respiration during this set amount of time. So let's write here R equals 2.2. Now remember, that's milligrams of oxygen per liter of water. And again, to get that, we took our initial dissolved oxygen concentration, subtracted from that the dissolved oxygen concentration after an incubation in the dark. Since only cell respiration was happening in the dark, that drop in dissolved oxygen was the amount of oxygen used by cell respiration. All right, another thing that this asks us to do is to calculate the gross primary productivity, the net primary productivity, and the gross amount of carbon fixed. So first of all, let's calculate the net primary productivity. That'll be the easiest one. We have an initial concentration of 8.4, an initial dissolved oxygen concentration of 8.4. We have a dissolved oxygen concentration for each bottle at each different amount of light. 100% of the light, 65% of the light, 25% of the light, 10% of the light, and 2% of the light. So remember, just like we did in the previous problem, to calculate the net primary productivity, we're going to take the amount of dissolved oxygen in our bottle in question, and we're going to subtract from that the initial amount. So for the 100% bottle, that'll be our first calculation. We're going to take our bottle DO reading, 10.2, and we're going to subtract from that our initial DO reading, which was 8.4, and that gives us a net primary productivity of 1.8. We'll repeat that same kind of calculation for the 65% bottle. So for that one, it's going to be 9.7 minus how much we started with, the initial amount, 8.4, and that's going to give us 1.3. And we'll continue that same pattern for our other bottles. So for the 25%, it will be 9 in the 25% minus 8.4 in the initial. So that gives us 0.6. For the 10%, 8.5 minus initial of 8.4, so that gives us 0.1. And for the 2%, we get 7.1 minus initial 8.4, so that ends up giving us a negative number, negative 1.3. You got to think about what that negative means. That negative means that with only 2% of the light, cell respiration is using more oxygen than photosynthesis is producing. Now let's calculate the gross primary productivity. To do that, there's a couple of ways we could do it. This says to take the bottle in question, so the 100% bottle, take its DO reading, 10.2, and subtract from that the dark bottle reading, 6.2. So 10.2 minus 6.2, we end up with 4. Now that's kind of a shortcut, the way that I would think about this, this problem. We talked about GPP, gross primary productivity, equals net primary productivity plus R. So we've already calculated for the 100% bottle that the net primary productivity was 1.8. We also calculated that during this amount of time, respiration uses 2.2 milligrams of oxygen per liter. So if we add those up, 1.8 plus 2.2, we get 4. So I'm going to use this calculation to get my answer. So for the 65% bottle, let's take the net primary productivity, which was 1.3. And let's add to that the amount of oxygen used up by respiration, which was 2.2. So 1.3 plus 2.2, we end up with 3.5. Complete that calculation for the 25% bottle. The net primary productivity was 0.6. The amount of oxygen used by respiration was 2.2. So that puts us at 2.8. For the 10%, the net primary productivity was 0.1. The amount used up by respiration was 2.2, so that puts us at 2.3. And for the 2%, negative 1.3 was the net primary productivity, plus the 2.2 used up by respiration, and we end up with a gross primary productivity here of 0.9. And again, that's milligrams of oxygen per liter of water. Now the final calculation is to calculate the gross carbon fixed. So again, it's asking for gross. So if we're going to calculate the gross amount of carbon fixed, we need to look at the gross amount of oxygen, the gross primary productivity. And again, to make these calculations, we have to use these equations from our AP formula sheet. So to do calculation 1, we need to take the gross amount of oxygen, which is 4, and I'm going to leave off the units here for space. Not a good habit to get to, but just for space, I'm going to take 4 milligrams of oxygen per liter. I'm going to multiply that by the conversion factor, which is 0.698. So 4 times 0.698. And that gives me 2.792. That answer is now in milliliters of oxygen per liter of water. All that top equation did is converted from milligrams of oxygen per liter of water to milliliters of oxygen per liter of water. Alright, so now we're going to take the answer from the top equation, 2.792, and we're going to plug that in here into the second equation. So 2.792 milliliters of oxygen per liter of water times 0.536. And again, that's just a stoichiometric conversion factor. And it's converting using the amount of oxygen produced by photosynthesis. It's going to calculate how much carbon was actually fixed or incorporated into the organic molecules of the photosynthesizer. So when we multiply that out, 2.792 times 0.536, we end up with an answer here of rounded to the hundredths place of 1.50 milligrams of carbon fixed per liter of water. And again, we're going to put that right here. All right, we'll continue that same pattern for our other calculations. So for the 65% row, we would take the gross amount, the gross primary productivity in terms of oxygen, 3.5, multiply that by 0.698, when we do that, 3.5 times 0.698, that gives us an answer of 2.443. And this answer is going to be in terms of milliliters of oxygen per liter of water. We're then going to take that answer, 2.443, plug it into the bottom equation, multiply it. So again, we're going to take our 2.443, we're going to multiply that by our conversion factor of 0.536, and that's going to convert us over from milliliters of oxygen per liter of water to milligrams of carbon fixed per liter of water. And we end up with an answer here of 1.31, which we will place right here in our table. We'll continue that same pattern for the rest of our calculations. So to do the 25% row, we would take our gross primary productivity in terms of oxygen. That's the 2.8 right here. We would multiply the 2.8 by our conversion factor of 0.698. We would then take that answer and multiply by 0.536, and that will give us a gross carbon fixed value of 1.05. Continue that same pattern for the 10%. Take our gross amount of oxygen produced, which in this case is 2.3. Going to multiply that by our conversion factor from our top equation of 0.698. We'll take that answer and multiply it by our conversion factor from the bottom equation, 0.536, to get a total amount of carbon fixed, a gross amount of 0.86. And then finally, the same thing on the bottom with the 2%. Take the gross primary productivity in terms of oxygen, 0.9, times the top conversion factor of 0.698. Take the answer of that calculation and multiply by 0.536 to get a gross amount of carbon fixed of 0.34. And again, that's milligrams of carbon fixed per liter of water. So here's a slightly more cleaned up version of that data table. We're going to use that to answer the next couple of questions. First question, using your data table, what seems to be the trend as the percent of light decreases and why? So notice as the percent of light decreases, as we go down, as the percent of light decreases as we go down, the amount of dissolved oxygen in the water decreases, the gross primary productivity decreases, the gross amount of carbon fixed decreases. So let's think about why there. The why to this answer is because there's less light, there's also less photosynthesis. Photosynthesis produces oxygen and fixes carbon. So as the percent of light goes down, there's going to be less oxygen produced, less carbon fixed. Question 2 is just the backward way of saying that same question. Using your data table, what seems to be the trend as the percent of light increases and why? So again, looking at the data table, as the percentage of light goes up, the amount of dissolved oxygen present goes up, the gross primary productivity goes up, and the gross amount of carbon fixed goes up. And the why to that question is the same as the why to the first question. As there is more light, there's more photosynthesis taking place. More photosynthesis means more oxygen produced, more carbon fixed. Question 3. Where would you say this organism is using about as much energy as they are making, and why? Well, first of all, making energy is kind of a bad way of saying it. You can't make energy, but maybe storing it might be a better way of putting that question. So let's go back to our data table. And what we want to look at is where does it seem to break even? Where is the net primary productivity closest to zero? And if you look at that, it's somewhere around 10%. Somewhere right in here, the plant is essentially breaking even. So that would be like if you had a job and you made a certain amount of money, and then the amount of taxes and other deductions were the same as the amount you made. Your check would be made out for essentially zero. And that's essentially what's happening here. This plant is doing photosynthesis, making organic molecules, and then it's using essentially the same amount of organic molecules that it made during the process of cell respiration. The last question in this section is, using your table and your graph, explain why most of the time there are bigger plants on land than in sea, and explain this in terms of evolution. Bigger plants need more energy. They need more organic molecules. So one of the main reasons that the bigger plants live on land is because there's more carbon dioxide present in the air than there is dissolved in the water. Plants have to have carbon dioxide to build sugars and to grow. Another reason is that there's more light present on land than there is in water. The water blocks out some of the light. Less light means less photosynthesis, which means less ability to make sugars, less ability to build big, large plants. All right, our last question in this video is part of an FRQ from several years ago. It says, in an experiment, net primary productivity was measured in the early spring for water samples taken from different depths of a freshwater pond in a temperate deciduous forest. So we're looking at a graph of net primary productivity versus depth. First part of the question that we have here, explain the data presented by the graph, including a description of the relative rates of metabolic processes occurring at different depths of the pond. So let's look at that part first. So notice that up here, in this part of the graph, the net primary productivity is positive. When you have a positive net primary productivity, that means that you're producing more oxygen, you're fixing more carbon than you are using either of those two. So in the top part of the graph, that should be telling you, hopefully, that photosynthesis is happening at a greater rate than cellular respiration. And that's because in the shallower parts of the lake, which is what we're looking at here, more light, lots of light is getting to those levels of the lake. Therefore, photosynthesis can happen fast. Both processes are happening, but photosynthesis is happening faster than cell respiration. Right in here, so somewhere around 20 meters deep, notice that the net primary productivity there is 0. That's the break-even point. At about a depth of 20, the rate of photosynthesis is essentially equal to the rate of cellular respiration. That means that plants are making as much sugar essentially as they're using. That's the break-even point. If we look at the bottom parts of the graph, depths below maybe 20 to 22, notice that the net primary productivity is negative. That should be telling you that the rate of cell respiration is greater than the rate of photosynthesis during this part of the graph. The plants are using more sugars than they're making. They're using more oxygen than they're releasing. So part C, that's what they wanted you to talk about. Now let's look at for a second at the rubric. Actually, we'll look at the rubric in another situation. What you should have said here, again, in the top part of the graph, the rate of photosynthesis is happening faster than the rate of cell respiration. Somewhere around 20 meters deep in the water, the break-even point, the rate of photosynthesis is about the same as the rate of cell respiration. And then if you go a little bit deeper than 20, maybe 22 to 23, at that point and deeper levels, cell respiration is happening faster than photosynthesis. And that's essentially because not very much light gets that deep. And without light, photosynthesis can't happen. Let's look at the last part of the question, part D. Describe how the relationship between net primary productivity and depth would be different if new data were collected in the midsummer for the same pond, and explain your prediction. So in the summer, we might be getting a little bit more direct sunlight, which means more light might go just a little bit deeper. And if that's the case, the break-even point, instead of being at 20, might be maybe at 30. So this graph might shift up a bit because there might be more light present. And we could also maybe get into the effect of temperature. That's another way you might approach this. But the way that I would approach part D is to say that in the summer there might be more direct sunlight, therefore more light makes it to deeper depths of the water. So the break-even point, the point where photosynthesis and cell respiration happen at about equal rates, might be at a little bit deeper level. All right, that sums up our discussion of dissolved oxygen and primary productivity. Hope that helped.